Hi everyone, today we are going to be looking at the brain. So first let's point out that we have a sheep brain here, which is much smaller than a human brain, and it doesn't exactly match our anatomy, but it's pretty close, so it makes a good replica for us to practice with. In the human form, the cerebellum and the brainstem exit the cerebrum in a more inferior manner, whereas in this sheep brain, you'll see that it's more of a posterior horizontal orientation versus a vertical orientation. First, we'll pick this up. We wanted to make sure that we know the difference between the dorsal aspect of the brain and the ventral aspect. So this is the dorsal aspect, where you can see the cerebrum, which is the largest part of the brain, and then the cerebellum, which is the second largest part of the brain. If we turn this over, this is the ventral surface, and you can see that there's a lot more going on, and we don't have that clear, beautiful view of the cerebellum. So we have a second specimen today that we're going to look at so that we can really properly see these meninges. So you can see how this is white in coloration. So that's telling us that this outer dura mater has not been removed from this specimen. So you can see here how it's loose here. It's not quite connected to the brain. And this is really thick and tough. I can pierce it with that, but to rip it, I'm really pulling hard right there, and I can't rip that. I can cut through it right there. So we have this beautiful dura mater. Now we have this very thin pia mater that is adhered directly to the brain, and the arachnoid matter fills the space in between them. And this is where the cerebral spinal fluid flows to bathe the delicate neural tissue. So now we can kind of peel that dura away to reveal the brain tissue underneath. And you can see how the dura is actually adhered pretty tightly. So what's breaking here, what you can see right here, is that arachnoid matter. And so we are separating that out. And then the pia mater is adhered to the brain. So my probe is under the pia. So remember, the cerebrum is the largest portion of the brain. The cerebrum has two hemispheres, the left hemisphere and the right hemisphere, and those are named for the side of the body that they're in while this is sitting in the living specimen. So the left side, or the left hemisphere, is on your patient's left side. So if you're looking directly at me or at this brain, your right side is the left hemisphere and your left side is the right hemisphere. The hemispheres are separated by the longitudinal fissure that you can see here in the center. And then we have another fissure, the lateral fissure, which is in this general vicinity. Again, sheep's brain, not quite exactly the same as a human brain. But the lateral fissure is going to separate the temporal lobe from the more superior parietal lobe. The first lobe we're going to look at is the frontal lobe, which you can see here on this most anterior portion of the brain. The frontal lobe is really important in thinking, planning, organizing, problem solving, all of those higher level, higher order functions that the brain does. It is also the seat of the motor control. So if we look in sort of this general area, we would find the central sulcus. A sulcus is a groove within the surface of the brain. You can see how the surface of the cerebrum is folded. The folds where they're raised are called gyri, or a singular gyrus. And in between the gyri, we have these grooves, which are called sulci, or a singular sulcus. So the central sulcus separates the precentral gyrus of the frontal lobe, which is important for motor control. So your motor commands are issued from the precentral gyrus. And then the postcentral gyrus of the parietal lobe, which is where all of your sensory processing takes place. To feel a sensation or become aware of a sensation, the information needs to reach this post-central gyrus. Sensory information is filtered by the thalamus, which we'll see when we do the dissection of the brain. But to become aware of it, it must reach the cortex. The parietal lobe, which is on this superior aspect of the brain, just posterior to the frontal lobe, is important for interpreting feelings as well as that sensory information in this post-central gyrus. The parietal lobe also processes taste, texture, and temperature. Moving inferiorly and laterally, we have the temporal lobe, which you can see here in this region. The temporal lobe is important in processing senses of smell, taste, and sound, and they also play a role in memory storage. And then this most posterior aspect is the occipital lobe, and the occipital lobe is important for interpreting visual input. So the optic nerves actually send information that goes eventually to the occipital lobe. So your sense of vision is processed in that occipital lobe. So if we move inferiorly, you'll see we have the cerebellum and the brainstem. But when we look at the cerebellum also has a folded surface, much like the cerebrum, but the folds are much smaller, and these are called folia when we are talking about the cerebellum. At the central, the cerebellum also has a right hemisphere and a left hemisphere, and in between the two hemispheres is this band of cortex called the vermis. So now we're looking at the ventral surface of the brain. This most inferior portion is the spinal cord. Just above the spinal cord, we have the medulla, which is important in autonomic regulation, like heart rate and respiratory rate. Above the medulla, we have the pons, and the pons is an important connection between the cerebellum and the brainstem. And it also functions in higher level control over the medulla, as well as visceral and somatic motor control. Just above the pons, we have the midbrain, which is this very small section, but the midbrain is important for processing sight, sound, and its associated reflexes. Just above the midbrain, we have the diencephalon, which is made up of the thalamus, the right and left thalamus on either side, the hypothalamus, which is the floor of the diencephalon, and the epithalamus is the roof of the diencephalon, which contains the pineal gland. The other structures we can see on this ventral surface are right here. This is where the pituitary gland was attached via the infundibulum, which is the stalk of the pituitary gland. The pituitary, if you remember, is housed in its own little bony case in the hypophyseal fossa of the sella turcica of the sphenoid bone. So when the brain is removed from the specimen, it is often stripped away. Just anteriorly to that, we see the optic chiasm. The last thing we can see here, this is the olfactory bulb. So I can decimate it completely.
Which happens a lot in dissections, folks.
So don't be afraid to
get in there.
Yeah, to really get in there. Okay, so that is the external anatomy of our brain. Now I'm going to dissect the brain so we can look at the interior structures.
Killing it.
All right, so I'm just going to cut right down this longitudinal fissure and then directly through the center of our cerebellum. And that is a beautiful section, if I do say so myself, which I do. So I'm going to move one out of the way so that we're just looking at a singular half here. So again, spinal cord, medulla, pons, cerebellum, diencephalon. So the cerebellum, you can see, is beautiful when you cut it open. It has what's called the arbor vitae, which looks like a tree. So the gray matter is on the outer portion or the exterior portion, and the white matter is in the interior. And you'll find that in the brain throughout, that the gray matter is exterior and the white matter is interior, which is opposite of the spinal cord, where the gray matter is interior and the white matter is exterior.
Also a great quiz question, folks.
It is. Some other things that we can see here are the corpus callosum, which is a band of fibers around this lateral ventricle that connects the left and right hemispheres and allows them to communicate with one another. The ventricle, we have 4 ventricles in the brain, the 2 lateral ventricles, so there's one here and there's one on this side, where you can see this very thin covering here, which sometimes gets demolished in the dissection process, but this is called the septum pellucidum. So it is this very, very thin covering to the lateral ventricle. On this side, what I'm going to do is put my probe in the lateral ventricle so you can see how deep and long it actually is. So the ventricle goes all the way back like that. The ventricles, you remember, are where cerebrospinal fluid is created in the choroid plexus. So the 2 lateral ventricles communicate with the 3rd ventricle, which is housed within the diencephalon, and then that communicates with the 4th ventricle in the brainstem, which is then continuous with the central canal of the spinal cord. That was our brain dissection. Thank you so much for joining us today.